Single-cell analyses revealed key tumor-infiltrating myeloid cell subsets associated with clinical outcomes in different subtypes of breast cancer published by Genes and Diseases. Breast cancer is the most common cancer and the leading cause of cancer death in women worldwide. Tumor-infiltrating myeloid cells, TIMs, key components of tumor microenvironment, TME, are considered to be potential therapeutic targets for cancer recently. However, their heterogeneity remains insufficiently characterized in different breast cancer subtypes. Here, we painted the landscapes of TIMs across breast cancer subtypes, and investigated the diversity of TIM subsets in different breast cancer subtypes. In different breast cancer subtypes from 10X and site dataset, we identified 5 common major lineages of myeloid cells, namely CDCs, PDCs, monocytes, macrophages, and mast cells based on canonical cell markers. In addition, CDCs, monocytes, and macrophages could be further divided into multiple subpopulations. In CDC subsets, except for the clusters of CLEC9A plus CDC1s, CD1C plus CDC2s, and LAMP3 plus CDCs, a discrete cluster of CXCL10 plus CDCs, which was previously not reported in breast cancer, was identified based on the expression of top marker genes in this cluster. Notably, expression of macrophage inflammatory proteins, PDL1 and its regulators were universally upregulated in tumor-derived CXCL10 plus CDCs compared to non-tumor-derived CDCs, suggesting that tumor-derived CXCL10 plus CDCs may have a capability of TAMS recruitment and cause T cells to differentiate into regulatory T cell, TREG, via PD-1, PDL1, thereby suppressing anti-tumor immunity in breast cancer tissues. Next, we investigated the developmental origins of CXCL10 plus CDCs in breast cancer. In tumor-derived CXCL10 plus CDCs, CD14 plus monocyte markers, S100A8, S100A9, and CD16 plus monocyte markers, CD16, were universally upregulated compared with CLEC9A+, CD1C+, and LAMP3 plus CDCs. Using Monocle 2, we found that the pseudotime trajectory began with monocytes in pre-branch and then split into CXCL10 plus CDCs in branch 1 or monocytes in branch 2, indicating a successful transition from monocytes to CXCL10 plus CDC. We next sought to explore the heterogeneity of macrophages in different breast cancer subtypes. Unsupervised graph-based clustering analysis gave rise to 7 and 6 macrophage subsets in breast cancer from 10X and site dataset, respectively. The top 3 marker genes in each subset as well as the proportion of each subset were shown in figure. Then, we further investigated the heterogeneity of mast cells in breast cancer. The mast cells in breast cancer were divided into FOSB plus and RPL31 plus subsets. Strikingly, ER+ and TNBC subtypes exhibited significantly higher RPL31 plus subset than FOSB plus subset, whereas HER2 plus subtype exhibited higher FOSB plus subset than RPL31 plus subset, indicating that imbalance of these two subsets might influence the prognosis of breast cancer patients. To explore it, we analyzed the DEGs of RPL31 plus subset compared to FOSB plus subset in breast cancer as well as mast cells in normal breast tissues, respectively. GO analysis showed that the down-regulated genes in RPL31 plus subset shown in Fig. S6b and S6c were majorly involved in negative regulation of apoptotic signaling pathway, suggesting that RPL31 plus subset might promote apoptosis of tumor cells and activate the immune response to limit pro-tumor effects of immune cells in breast cancer, whereas FOSB plus subset might have an opposite effect. This might be the reason that imbalance of these two mast cell subsets affect prognosis. In conclusion, our study illuminated key TIM subsets associated with clinical outcomes in different subtypes of breast cancer patients. Our data presented here may help identify better molecular targets for prognosis and treatment, and provides new insights into the immunotherapeutic strategies most appropriate for each subtype of breast cancer.